Good morning, everybody. Together with my co-moderator, Kavisha Arora from Cedars Sinai Medical Center in Los Angeles, I would like to thank you for your presence here, for attending this session. This workshop, number 4, will be focused on emerging topics in CFTR cellular biology. As you are well aware, we have almost 2,000 different CFTR mutations that cause the dysfunction of the CFTR proteins by acting in many different ways. We have 6 or 7 different classes of mutation, but fortunately, during the last years, 4 different drugs have been developed that can rescue the CFTR dysfunction caused by many of these mutations. We have 1 potentiator monotherapy, and we have 3 combination therapies consisting of 1 or 2 different correctors plus a potentiators. Together with these 4 different drugs, we can functionally correct approximately 180 different CF-causing mutations, accounting for 90% of individuals with cystic fibrosis. However, this percentage can differ in other countries. For example, in Italy, we can treat more or less 70% of CF people. In addition, we can see that the clinical benefits are variable in different patients. Adverse events have been reported, such as chest tightness, diarrhea, and drug-drug interaction, which are not the ideal since we are talking about lifelong drugs, lifelong treatments. Among the mutations that cannot be treated by the already existing drugs, we have approximately 10% of mutations that cause the generation of premature termination codon. So the PTC mutations are a major issue for among the yet untreatable mutations. What's the next? What are the next focuses, aims of CF basic research? First of all, we have to develop novel small molecule therapies, in particular for PTC mutations. We need to develop gene editing techniques, but we need also to increase our knowledge, our understanding of CF cell biology. Because in this way, we could identify, for example, alternative targets that will be crucial for the design of better combination therapies. They will be important to target the refractory CF mutations, the ones that are not treated by existing drugs. And we need also better drug to address the CFTR-independent CF comorbidities. So the goals of this workshop will be essentially 3: to evaluate the progress towards developing small molecule therapies for PTC mutation in CFTR, to assess the therapeutic potential of CFTR gene editing strategies, and to analyze the potential alternative targets to restore CF airway epithelium homeostasis. To focus on our aims, we will have 5 different talks that you can see summarized here. And I hope you will enjoy these different talks dealing with emerging topics in CF cell biology. So our first speaker.
Good morning. My name is Kavisha Arora. I'm from Cedars Sinai, and I welcome you all to this workshop. So the workshop consists of 5 presentations, 15 minutes each, followed by 8 minutes of discussion. So I would like to introduce our first speaker, Jian Guo Chen. And he will be presenting on synthetic aminoglycoside ELX02 induces read-through of CFTR G550X, producing superfunctional protein that can be further enhanced by CFTR correctors. Thank you.
Where's my... Oh, thanks. Good morning, everyone. Today I will talk about synthetic aminoglycoside ELX2 induces read-through of CFTR G550X, producing super-functional protein that can be further enhanced by CFTR correctors. This is my disclosure. As we know, there are no approved therapies for premature termination codon PTC mutations, which affect about 11% of CF patients. The aminoglycoside ELX2 promotes the read-through of PTC bearing CFTR transcripts by allowing the insertion of near-cognate amino acid tRNAs. In an in vitro study, G550X CFTR intestinal organoids were more responsive to ELX2 treatment than G542X CFTR organoids. However, it remains unknown why G550X CFTR is more responsive to read-through treatment and the role of CFTR modulators for these mutations. In present study, several approaches were used, such as G550X CFTR mutation compared to more common G542X mutation upon treatment with G418 or ELX2. FIS assay to measure functional response in patient-derived intestinal organoids. A TurboGFP report system to determine amino acid inserted during read-through in CFTR PTC suppression. FRT cell lines stably expressing G550 variants generated based on a Philippine system. Read-through and CFTR function measured by TEC assay. CFTR protein expression determined by West blot. And patch clamp and TEC assay to identify the mechanism of differential response to read-through treatment. This slide showed the forskolin induced swelling response to ELX2 in G550X CFTR or G542 CFTR intestinal organoids. On panel A, a representative swelling curve showed the G550X organoids had a ELX2 dose-dependent increase in swelling over time. On panel B, G550X organoids had a significant high swelling response to ELX2 compared to G542X organoids, indicating that G550X allele is more responsive CFTR allele to read-through treatment. We want to know why G550X CFTR are more responsive to ELX2 treatment. As we know, the amino acids inserted at PTC upon read-through are largely context-dependent. Resulting missense CFTR mutations can affect the processing and the function of read-through CFTR protein. We hypothesize that amino acid inserted in the G550X position could lead to increased CFTR function. To test our hypothesis, we used a TURP-GFP report system to determine the amino acid inserted during ELOX2-mediated G550X PTC separation in HEC cells. This report includes TURP-GFP, CFTR, cassette, which includes G550X, UGS stop codon, and local context, including 3 codons of upstream and downstream of the stop codon. Read-through report, HA tag, and protein purification mark, his tag. So after PTC separation, the full-length protein will contain TGFP along with HA and his tag, whereas the premature termination protein will contain TGFP only. Western blot confirmed that increase of the read-through protein, as shown in the yellow and the green bands in ELOX2-treated HEC cells. Mass spec analysis of read-through proteins found that tryptophan was the sole amino acid inserted in the G550X position, which was different from previous findings in G542X PTC separation with 3 near-cognate amino acids. As we know, the amino acid tryptophan differed from the amino acid glycine, normally in the wild-type CFTR. So we want to know whether the function of the G550W CFTR protein is effective as the wild-type CFTR. We generated the FRT cell lines stably expressing G550W CFTR. As you can see on the panel A, compared to Y-type CFTR, G550W CFTR protein expression level was similar. On panel B and panel C, the G550W activity was significantly higher than Y-type. So this data indicating that the insertion of the amino acid tryptophan in G550W CFTR augments CFTR function without altering CFTR processing and trafficking. To understand why G550W CFTR activity was significantly better than Y-type CFTR, we investigated the Fosclin dose response for CFTR activity in G550W and Y-type FRT cells. Compared to Y-type, G550W CFTR had a significant left shift of Fosclin dose response curve. On the panel B, you can see that, indicating that G550W CFTR is more responsive to Fosclin than Y-type CFTR. Using macropatch excited from HEC cells, we performed pKa titration in G550W CFTR and Y-type CFTR. As you can see on the panel C, compared to Y-type, G550W CFTR had a significant left shift of pKa dose response curve, indicating that G550W significantly increased pKa sensitivity, leading to enhanced CFTR channel activity. The single-channel patch clamp excited from FRT cells found that G550W CFTR channel increased channel open probability on panel C, and this was supported by increased mean burst duration MBD on panel D and reduced the interburst interval IBI on panel E compared to Y-type CFTR. Furthermore, we evaluated whether CFTR correctors VX445 and VX601 can augment CFTR protein expression and function in G550X or G542 FRT cells following G418 or ELOX2 treatment. As you can see, compared to vehicle, G418 or ELOX2 alone treatment enhanced the CFTR protein expression in both FRT cells. Additional correctors further augmented CFTR protein expression. We also found that the CFTR protein expression level in G550X FRT cells was significantly higher than G542X FRT cells. Moreover, consistent to the protein expression, CFTR function in both FRT cells was enhanced by G418 or ELOX alone treatment, and further augmented by additional CFTR correctors. And the CFTR function of G550X FRT cells was significantly high compared to G542X FRT cells. Particularly, in G550X FRT cells, G418 or ELOX2 alone treatment enhanced CFTR activity reaching about 20 and 10% of Y-type levels. Additional correctors further augment CFTR activity reaching about 40 with G418 or 20% with ELOX2 or Y-type level, respectively. In conclusion, compared to more common G418, G542X PTC mutation, G550X has a great response to aminoglycosides due to the gain of function property of the CFTR read-through product. This was explained by the insertion of tryptophan in the G550X position augments CFTR channel forscaling and PK sensitivity and open probability. In combination with the clinically approved CFTR correctors, the function of G550X CFTR can be further improved. G550X may be a particularly sensitive target for translational read-through therapy and may be emblematic of other super-responder read-through mutations. At last, I would like to thank UAB CF Research Center, Dr. Steve Lowe and his lab member, Dr. David Bedwell and his lab members, Elux Pharmaceutical Company and Hub for their great help, support, and contributions. Thank you.
So this session is now open to questions. Yeah, I'm a little confused. Actually, I think we are supposed to type in the questions. Right?
I think you, okay.
Yes, we have G542X, we have 1 homozygous and 5 or 4 heterozygous. Yes. Mm-hmm. Right. Yeah, great questions. Based on the reference, as we know, the G550E can restore part of the delta F functions. And since the E is different from W, the tryptophan, so the structure is different. And although the local context is similar in the SGQQ, the motif area. So it's possible if you change it from E to W, maybe it's better. We'll never know, but we can try to test that. Okay, thanks. Thanks. Good question. Currently, we tested the UGA stop codon, but we never know the other stop codons. So right now, we have not tested, but it's deserved to do that. Thank you. No. Okay, thank you. Yeah, I think this is a very good idea. Maybe in the future we can try. Yes, thank you. That's true. You're right. Completely right. Thank you. Yeah.
I think we will move to the next session because the time is up. So any other questions that can be followed up after the completion of the workshop? So we'll move to the next speaker.
The second speaker is Martin Ondra. CRISPR-based cellular models with endogenous expression of EBIT-tagged wild-type and mutant CFTR enables high-throughput biology studies and screening for new transporter modulators.
So good morning, my name is Martin Ondra. I'm a PhD student at the Institute of Molecular and Translational Medicine in Czech Republic. First of all, I would like to thank the organizer for giving me the chance to be presenting here our work at NACFC. Today I'll be talking about the preparation of new cellular model for the cystic fibrosis and drug discovery. There are no relationship to disclose to that presentation. So there is emerging need for discovery of new CFTR correctors and potentiators. High-throughput screening is a sophisticated way to help to discover such drugs for patients suffering from cystic fibrosis. There are two main approaches in the high-throughput screening to discover such drugs. The first one, phenotypic assays, and the second one, direct protein localization assays. In our lab, we have decided to prepare a new model for the direct protein localization. So we use the CRISPR-Cas9 technology to insert the tag and tag the CFTR with the endogenous expression. So it was crucial to preserve the CFTR trafficking and function. So it's important to choose the right tag and also the position. We have decided to put the tag inside the fourth extracellular loop because it was published several times in different journals. So that was the position which we have chosen for in introduction of tag. Our main aims was to establish a relevant cellular model for HDS with the endogenous expression of wild-type CFTR in bronchial. Should I try without? Because I'm pretty loud. This one? I don't know. I can do it without?
Again, yes.
Yeah? Okay. So let's continue. Okay. So our main aim was to establish a new relevant cellular model for HDS, which will have the endogenous expression of wild-type CFTR in the bronchial epithelial cells, which could be used for fast detection of changes and also its localization in the plasma membrane, and mainly it could be used for high-throughput screening of chemical libraries. Maybe you would ask why you are tagging wild-type CFTR. It's a healthy protein. So that's the reason why we are doing that, is that we want to prepare the pipeline and always has the same genetical background and start with the wild-type CFTR. So we will create a building block starting from wild-type CFTR for future insertion of pathogenic CFTR mutation inside the wild-type CFTR. So that's the rationale behind that, why we are working with the wild-type CFTR firstly. So as I told you in the beginning, we are tagging CFTR. And the tag which we used in our work was a hybrid tag. It's 11 amino acid peptide tag, which was developed from the C-terminal region of nanoluciferase by Schwinn et al. So this small peptide, it doesn't work as an enzyme. There is also the counterpart, the bigger part, the rest of the luciferase, which is called larger bit, and it also doesn't work as an enzyme. So when these two counterparts are getting together, closer in the proximity, they couple together with a non-covalent bond, and the function of the enzyme is restored. So you provide a substrate, and you can measure the luminescence. So we use the CRISPR-Cas9 technology to insert that tag in the fourth extracellular loop to be able to monitor the endogenous expression levels and also its localization in the plasma membrane. My talk today will be divided into two parts. I will start with the wild-type CFTR targeting to show you how we design all the CRISPR-Cas9 technology, what we did for the optimization steps, and in the other part, I will show you all the clones preparation process. So let's start with the wild-type CRISPR targeting. We designed a couple of CRISPR RNAs to target the fourth extracellular loop. We use the ChopChop online tool, which is available, and designed the CRISPR RNAs for us. And we have decided to put the tag in the two different places in that fourth extracellular loop. So the first place was before the glycosylation, and the second one was after the glycosylation. So you see that we designed four different CRISPR RNAs. So to start from the scratch, we wanted to know that we are starting with the good delivery system. Can you hear me? Okay. So we designed 4 CRISPR RNAs, and we wanted to know that we're starting with a good starting point. So we tried if the lipofunction is good, if the electroporation is better. So we found out that the lipofunction was not good. So we stick out with the electroporation to deliver the RNP complex, which is a Cas9 enzyme with a guide RNA to target the fourth extracellular loop. So as you can see in the results, we were good in the electroporating the cells and getting the RNP complex, which is the CRISPR RNA together with the tracers is creating guide RNA, and the guide RNA is labeled with the fluorochrome. So when you have the RNP complex, Cas9 plus guide RNA, so you are getting into cells and you see that these cells are electroporated. So we did the confocal microscopy, also we did the flow cytometry to confirm that we are starting with the good electroporated cells. Also, we used a type program, which is an online tool, to estimate the cutting efficiency of these CRISPR RNAs. As you can see, we continue with the CRISPR RNA number 1 and CRISPR RNA number 3. Both are targeting different regions in the CFTR. Because these were cutting more than 50%, they had more than 50% cutting efficiencies. As I mentioned, we continue with the CRISPR RNA number 3 and number 1. Like to get the hybrid tag inside the CFTR, we needed to use the homology-directed repair of cells. So we designed two different SSODN templates, or single-stranded DNA templates, to use it for homology-directed repair and knock-in of hybrid. So we created 4 different conditions with the different length of linkers, because we worked with the hybrid previously, and we knew that sometimes it could be tricky when this like in a rigid structure, so it's better to be like sticking out from the loop. So we designed 6 amino acid and 8 amino acid linkers on both sides of hybrid to be more accessible for the large bit. And we designed these 4 different combinations. We tested them with 2 different approaches. First one is the lytic assay. In this assay, you do the lysis of cells, you release all the protein tag with the high bit, you provide the large bit and also the substrate, so you can measure the total level of protein which is in the cells. So you can see we compare these 4 different conditions, and it proved that the 8 amino acids are better and providing a higher signal. There was also nice correlation between the seeding number of cells already in the bulk culture, because this was tested on bulk culture, so not every cell was modified by the CRISPR. We already see a nice correlation in seeding numbers. We did the same thing for the extracellular assay. This assay measured the number, or like the amount of CFTR in the membrane. So it's done on the living live cells. So there is no detergent to do the lysis of cells. You provide a large bit substrate and you measure the level of CFTR in the membrane. And we saw the similar trend that the 8 amino acid linkers are better than the 6 amino acid linkers. So we continue only with the condition on both sides of for the hybrid before or after glycosylation, but with 8 amino acids. So we excluded the 6 amino acid condition for preparation of clones. So let's start with the clones preparation. We use the limiting dilution assay to prepare the clones. We prepare more than 182 clones, and we start with the primary screening to identify the positive clones for the hybrid. We use the lytic assay to do the lysis and see the total level of protein, and we identified 32 clones positive for the hybrid. These clones were retested in the secondary screening, combining the lytic and extracellular assay to see if there is protein in the membrane. So we got the various level of signals in the extracellular assay. So we decided to do the PCR phenotyping to distinguish the homozygotes and heterozygotes. So we got 9 homozygotes and 18 heterozygotes in this assay, and we sequenced only the homozygotes. And here in the figure E, you can see a nice example of a knock-in of a hybrid with 8 amino acid linkers from the sequencing traces. When we got these clones, homozygotes clones, we started validated these clones by different assays. So we used the Western blotting to see if the protein is glycosylated, what the level of protein. Also, we did the hybrid blotting, which is the similar assay to Western blot, but there is no immunostaining on the membrane. It's just using the large bit instead of primary and secondary antibody, so you can read the luminescence on the membrane. We also did the immunocytochemistry to see if the protein, like another independent assay to see if the protein is in the membrane, and we tried without and with permeabilization. And that was the antibody against the first extracellular loop of CFTR. And we see a nice signal in the membrane without the permeabilization, and we see a nice signal with permeabilization inside the cells. We also did the transepithelial electrophysiology using chamber method, and we see a nice response to forskolin and also to inhibitor of CFTR. And lastly, we started with the optimization to transfer our assay to 384 well-plate format, because we want to use it for our HTS screening platform, and we wanted to know what's the time window to use it for that assay, and also we wanted to see what's the homogeneity of the assay among the wells in the 384 well plates, and it was pretty nice. So to conclude, we prepare the new model for high throughput screening for now with the wild-type CFTR, and this building block will be used in future for insertion any other pathogenic mutation, CFTR mutation, to create other models which could be used for discovery of new drugs for patients with cystic fibrosis. And also I promised you that I'd like to explain you why we started with the wild-type CFTR, because we tested all the methods to prepare the clones, to validate them, also what we will do for the optimization of CRISPR. So we created for our institute a new pipeline, which we'll use in future for insertion CFTR mutations into the wild-type CFTR. So I would like to thank all my colleagues for working with me on this project, and also thank you for your attention.
Thank you very much, Martin. Any question about this talk? Okay.
Yeah. Yeah, it could be used for that. Like you need beads, and like there is an antibody against the hybrid tag, so depends. Like also there's a large bit binded to the beads, so you can do also pull down assay on that, with that. You mean like a preparing, or like how efficient is to isolate the protein with the like in simple? We just started with the pulling down assay, so I cannot tell you the exact numbers now. Yeah, but like the greatest benefit is the size of tag. It's really small, so it's not affecting the whole molecular weight of the protein because it's really small. And also we chose the Fortex loop because we found in the literature that like people are using that loop, and it can handle other modifications.
So if there are no other questions, no, okay.
I'm sorry, I cannot hear. About what?
We can ask you later.
Later. Yeah. Yes. Mm-hmm. 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 Yeah, it was that way all the time. We did like 3 biologic replicates, it's just one of many replicates which we did, but I'm not qualified to answer this question because I'm not the guy who's doing the electrophysiology. But like I would like to get like we tested only these 2 because it was not done in our institute, so we sent these 2 clones only for confirmation because we knew that these 2 are homozygotes responding in the other assays, so we tested these 2 clones from 8 or 9 homozygotes which we got. Well,
I have one question, if I missed. What cell are these?
HB human 16 HB 14O human bronze. Yeah.
Thank you. We'll move forward to the next presentation. Thank you, Martin. Our next presenter is Celeste Ripe, and she's going to present on genome-wide functional genomics analysis of F508 CFTR degradation. Thank you.
Hi, everyone. Ooh. Okay, I'm going to set that back. Can everyone hear me okay? Okay, great. I'm Celeste Ripe. I'm a postdoctoral fellow in the Computo Laboratory at Stanford University. And I have, whoop, where's the thing? Okay.
Try to use the arrow.
Okay, the arrow, and then, okay. And I have nothing to disclose for this presentation. So today I'm delighted to talk to you guys about my work to conduct genome-wide functional genomics analyses of F508 delta CFTR degradation. So the central question that my work is trying to answer is, what is the quality control machinery that recognizes misfolded F508 delta CFTR? Now, as CFTR is translated at the ER membrane, it folds, and then the cell realizes, ooh, this protein is misfolded, so it recruits triage factors that then send it off to the ubiquitin machinery, which tags the protein with ubiquitin, and this is through the activity of E3 ubiquitin ligases as well as E2 ubiquitin conjugating enzymes. And this signals the cell to dislocate CFTR from the ER membrane and send it off to the proteasome to be degraded. This process is collectively known as ER-associated degradation, or ERAD. Now, CFTR has been characterized as an ERAD substrate for about 28 years, but the exact mechanism of how this works is pretty murky, and there's a lot of conflicting studies out there in the literature. For example, the E3 ubiquitin ligase, you can look at the literature, and there's at least 5 different E3 ligases that have been implicated, including RNF5, HERD1, CHIP, GP78, RNF5, 185. So the question is it some of these E3 ligases? Is it all of them? You know, what's going on? So in order to sort of get in to answer this question, I decided to adapt a genome-wide CRISPR-Cas9 screening platform developed in the Capito laboratory. And what the screening platform is that it takes a transgenic, fluorescently tagged F508 delta CFTR, integrates that into the K562 cell genome, and then we add Cas9 BFP, and then we infect with a guide RNA library that targets all 20,000 genes in the human genome. And there are about 10 guides apiece. So this is about a 250,000 guide RNA library. We then induce CFTR expression with doxycycline. We wash out the doxycycline to induce a chase. This causes the protein to degrade over time. And then we do fluorescence-activated cell sorting, and we gate for the top 5% fluorescent cells. And then we sequence the guide RNAs from the sorted population and compare that with the guide RNAs in the unsorted population. And then we can calculate a gene effect source. So we have some hits that are stabilizing. Those are well enriched in our sorted population. And then we have some hits that are accelerating, which are disenriched in that top 5% population. So our assay really depends on a really nice reporter cell line. And so K562 cells do not endogenously express CFTR. So we want to make sure that CFTR biology is behaving as expected in this cell line. So what we can see is the transgenic F508 delta reporter, which is tagged with M neon green, which is like GFP, only a bit more stable and brighter. We see that it is, in fact, in the band B form, the ER-associated form. The wild-type construct was found in both the band B and the band C form. Of course, the band C form is the plasma membrane-associated form. Then we did a kinetics-based assay. So I'm going to go through this in bit detail because I'm going to show a lot of kinetics-based assays. So what I do is I express CFTR, and then I inhibit translation with emetine, which is like cyclohexamide. And that stops translation, and we watch the protein degrade over time using flow cytometry. So what I'm showing you here is wild type versus F508 delta. And you can see that the F508 delta really decreases the protein half-life of CFTR. It's the half-life in K562 cells is about 35 minutes, so that takes 35 minutes, half the molecules are gone. In contrast, wild type is much more stable. We've got a little bit of degradation at the start for some wild type CFTR that misfolds as it's translated at the ER membrane, but then it has a nice stable population. Another thing we did as part of the validation goes is when we wanted to make sure that CFTR was a target of the ubiquitin proteasome system. So just as a refresher, the way the ubiquitin cascade works is that ubiquitin activating enzymes, E1s, recruit ubiquitin, activate ubiquitin using ATP. This is then transferred to the active cysteine on the E2, and this ubiquitin is conjugated to the E2 is recruited to an E3 containing the substrate. The substrate is then ubiquitinated, and this ubiquitinated protein goes to the proteasome where it's then degraded into the constituent amino acids. So we looked at 2 steps in this cascade. We looked at the E1, we inhibited this with TAC243, and then we looked at the proteasome, which we inhibited with bortezomib. What we found is that TAC243 completely stabilized CFTR. It's a flat line. However, bortezomib only stabilized most CFTR molecules, but not all CFTR molecules, suggesting that there is some ubiquitin-dependent but proteasome-independent degradation of CFTR. Now, we conducted our genome-wide screens, and we saw that RNF5 was the top E3, and we saw that UBE2D3 was the top E2, we got about 45 E3 ligases that came out of this screen. We got about 1,000 significant genes, but to be frank, most of these are just transcription and translational genes. They sort of, they modulate the expression of my reporter, but it's not clear if they're involved with the degradation of CFTR. So we looked at this data set, and what actually really struck us was how, okay, sorry, this is, sorry. Okay. Okay, I don't know what's going on here. Sorry. Okay. I'll just use the laptop.
Yeah.
Okay, I'm going to set that thing. What really struck us in this screen is that F508 delta CFTR had fewer strong confident hits than our model ERAD substrates. So my lab has done a lot of CRISPR-Cas9 screens with a lot of different ERAD substrates. And when you look at the model ERAD substrate, where we really know the pathway and everything's great, the ERAD genes just pop right out of the screen. What you're looking at the y-axis is the Castle score, which is a measure of confidence. Castle effect is the measure of enrichment. But what you see with CFTR is the effects are much more modest. And so this led us to hypothesize that actually our screen hits only have very minimal effects on CFTR F508 degradation. And so when we knocked out our top E3 ubiquitin ligase, RNF5, it only had a modest impact on half-life. So what you're showing you is there's an emetine chase experiment with 3 different guide RNAs against RNF5. The full change at half-life, the maximum that I measured, was only about a 2-fold change. This is taking the CFTR half-life from 35 minutes to about 70, which is very different than the 10-hour half-life of wild-type CFTR. We also observed is that we mostly saw an increase in band B. We also saw a modest increase in band C, but we saw no change in the ratio of the two. We also knocked out the top E2, UBE2D3. We got the exact same result. Very modest changes in CFTR degradation rate, and we saw, although this is very difficult to see in this Western blot, a very modest increase in band B levels, very little rescue of band C. So UBE2D3 has never been characterized as the E2 of RNF5, so we wanted to do the double knockout experiment. And to our surprise, these actually acted in completely separate quality control pathways. So if you knock out the genes separately and then we look at the double knockout, the effect is pretty much additive. And again, we see that we're rescuing band B in the double knockout, and we're getting a bit more band C, but we're not increasing the ratio of band B to band C. So another question that we were interested in asking is, do correctors abrogate the activity of RNF5 or UBE2D3? These are clearly the top 2 genes in the genome-wide screen. Are correctors acting through this mechanism? So we looked at 3 different classes of corrector drugs. We looked at type 1, type 2, and type 3 correctors. So type 1s are thought to bind transmembrane domain 1, type 2 thought to bind NBD2, type 3 thought to bind NBD1. And we first wanted to see that if our kinetic model recapitulated a lot of the functional data that we know about these drugs. And so what you can see is that VX661, tezacaftor VX445, both had modest impacts on CFTR stability, but then when you combine those 2 drugs, you had a really strong synergistic effect. Then we looked at what happened when we added Core4A to the mix, and indeed we got a little bit increase in CFTR F508 delta stability. So then we performed gene-drug epistasis experiments. So there are 3 results that you can get for gene-drug epistasis experiments. So one is additive, which is where the gene and the drug act in independent pathways, so they add together. Synergistic is when they're in compensatory pathways. So if you add the gene and the drug together, and by gene I mean a knockout together, you get a nice synergistic effect like what we would see with Trikafta. And then buffering is the same pathway. So if we knock out the gene, we add the drug, there's really no change. And what we saw is that corrector drugs act in pathways that are independent from RNF5. So if you knock out RNF5, you add in the corrector drug, you see an additive increase. And this is true for type 1 correctors, type 2 correctors, and type 3 correctors. This nicely complements some data from the Pedamonte lab. We looked at type 1 and type 2 correctors at a functional level with CFTR. And we did the exact same assay with UBE2D3 knockouts, and we got the same thing. So I've now shown you that we have 3 drugs acting in one pathway, or in 3 separate pathways, and I've shown you that we have 2 genes acting in separate pathways. So what happens when we put all 5 together? And so what you can see here is that we have the no drug in black, we've got the double knockout in purple, we've got the 3 drug combination in brown, and then what we see in maroon is all 5 stimuli together. And what we see is an 8-fold change in half-life. This is by far the strongest change in half-life I've seen. Somewhat it's crazy to me that this 8-fold change in half-life is what I would see with like a model ERAD substrate with 1 guide RNA, but this I have to add 2 guide RNAs and 3 different corrector drugs. So in sum, CFTR is degraded through at least 5 different ubiquitin-dependent quality control pathways. So we can think of the type 1, type 2, and type 3 mechanisms of correctors that we all know, but we can also think of abrogating RNF5 and UBE2D3 as separate mechanisms for correcting CFTR. So with that, I'd like to acknowledge my laboratory, the Capito Laboratory. I'd like to thank my collaborators in the Bassett, Gittler, Weiss-Corey, and Mio laboratories, as well as my home away from home, the Stanford Shared Facts Facility, as well as my funding sources, the NIH and the CFF.
Thank you, Celeste. This session is now open to questions, please.
Hi.
Fascinating. So, were all the chases done in K563s also?
Yes.
So, I know it's great for the SG screens.
Yeah,
Leukemia cell line.
of course.
Have you ever gone back to something that maybe the field uses more commonly, right, to the guide RNAs and see if you see other effects?
Yeah, so we are going, that's in the works, it's just not completed yet. Yeah. Hi, Eli.
Excellent. So this slide where you compare a model of ERAD substrate to CFTR, you see like a lot less ubiquitous. Are the E3s and E1s identified with CFTR, are they all associated with ERAD, or are some of them associated with cytosolic degradation machinery? The CFTR has 3 cytosolic domains, but I feel like we don't think a lot about the cytosolic machinery going on in the ER.
Yeah, so actually RNF5 was the only ERAD E3 that identified. The E2 that I identified is cytosolic. But most of the E3s that I showed were actually involved in transcription and translation, so I think they're just having these modest effects. And to be honest, my screen is pulling out these really, really small effects. Like UBE2D3's effect on CFTR is like 30% at the bulk level. It's very small. Hi. Yeah, those were translation. So my assay involves the degradation of a transcriptional reporter. And so basically I wash out doxycycline, I have an unstable RNA, that mRNA decays, and then also the protein decays. And those factors are the XRN1, XRN2, which are important for that degradation. Hello. Yes. Yes.
So my question is, if you go back to the original case to see whether there is no additivity in some of the drugs you tested?
No, I have not done that yet. Right now, what I'm actually working on are sensitized screens where I do a genome screen in the RNF5 and UBE2D3 knockouts, and I'm also doing screens with the corrector drugs as well to try to get at those 5 independent pathways. Yes?
I think I just remembered that RNF5 actually acts as a chain ligation, right? Acting after 78.
I think it acts as, I can't remember off the top of my head. I thought it was just an E3 and it wasn't.
Well, it is an E3, but some add the progenitor ubiquitins and they're elongated.
Yeah, yeah,
I think that was from the Gada's lab.
okay.
If that's true, right, then what could be happening is that elongation is giving a subtle effect, but it's the primary, the third one, and the 78 is making it this issue.
Yeah,
I actually think about, even though you don't see those as a redundancy, we want to actually go back and think about it.
yeah, no, that's a great point, and I'm really hoping that the sensitized screens will pull out some of those hidden E3s that I'm not seeing in this initial screen. And also, I should say, I'm giving a poster after this, so if people want to talk more, I'm at 656.
I have one question. Have you identified any ubiquitination pathway that removes the peripheral CFTR, not the ERAD?
No, I have not. I did look for that, but I don't think I'm getting very much going to the peripheral. I'm getting maybe like 10% that goes out to the peripheral. So I need to, basically, I either need to do the assay where I do it at 30 degrees, or I need to like knock out the ERAD E3s to look at it. Yeah.
Thank you, Liz.
We can move to the next speaker, Shuyu Hao, a strategy to edit disease-causing segmental duplication in CFTR.
Oh, sorry. Can you hear me? Hi, everyone. I'm Xu Hao. I'm a postdoc from Mitch Drum's lab at Case Western Reserve University in Cleveland. Oh, sorry. I have no disclosure today. So, as my title alluded, this study was designed to correct segmental duplications in CFTR. Duplications belongs to copy number variants or CNVs, which includes both duplications and deletions of a big segment of DNA sequences. So CNVs are not rare, depending on the stringency. About 5 to 10% of genome contributes to CNVs. A separate study investigated intragenic CNVs, and those CNVs were observed in about 10% of individuals who have clinically significant result. So those studies tell us that CNVs are pretty frequent, and many of them, they are pathogenic. However, mostly due to previous genotyping tests, CNVs are not very well studied compared to other variants like single point mutations and small indels. Same as in general, CNVs are understudied in CF as well. As we just learned, about 180 mutations have been taken care of, but CNVs, as far as we know, there is no therapeutic ways to address those variants. We started this project when there was a patient referred to my PI, Dr. Drum, by her physician. This patient who carries segmental duplication in one of her CFTR alleles. So here I'm showing her the schematic of her mutation and our proposal to correct it. So this is her CFTR gene. As you can tell, up to, this is both introns and exons. Up to exon 6, everything is fine, but she has 2 copies of exon 7 to exon 11, as shown here. So we have been working with CRISPR-Cas in the group at CASE, and we, based on our experience, we proposed this pretty straightforward but potentially very efficient way to remove this extra copy of CFTR DNA sequences. So we just need one single guide RNA which target, it could be any region, this region, but we try to avoid any disruptive effect of the gene. So we design our guide RNAs in introns. So we just need one guide RNA, but which will cut twice because of the duplication. So any sequences from the first cut, before the first cut, and after the second cut will remain, but anything at the DNA sequence in between will be removed by this guide RNA. That will give us a normal copy of CFTR gene as we designed. And there are many well-studied mechanisms that are responsible for segmental duplications. And in this case, we think, and mostly, the duplication happens during meiosis. There are 2 alleles of chromosome genes, 2 alleles. They misalign during meiosis, and crossover could happen, and recombine those alleles equally. And this will lead to duplication in one chromosome, and the recipient chromosome will have a deletion, big chunk deletion DNA sequences. So in this project, we only focus on segmental duplications, not deletions yet. And we looked over, and there are many disease-causing CFTR duplications have been identified in CFTR gene. And this is those the lines, the horizontal blue lines represent the locations and the sizes of those duplications. Some are bigger than the others, as you can tell. Even the smallest one is over 1 kb. If our strategy works, it can be beneficial for a big group of patients who now have no cure. So right now, we are focusing on the CFTR dupe 7 to 11 duplication in the lab. Sorry, so I forgot to tell you that this duplication, the one we are focusing on, the CF 7 to 11, which is 27 kilobases, it's very, very big. So in theory, we can design many guide RNAs within this region, even in introns. So we need a reporter system to tell us if those guide RNA, they can work efficiently to make cuts and make the deletion as we wanted. So the reporter system we have, as shown here, we have 2 separate plasmids. One is called dupe reporter, and we have guide RNA targeting sequences on both sides of this 3 poly A transcription terminator. And we also have LOXP sites for CRE to target as control. So this reporter has M-scarlet as a fluorescence marker, and we also has a luciferase as a bioluminescence marker. When the other plasmid encodes guide RNA, Cas9, and EGFP. So when those 2 plasmids co-transfected in the cells, the guide RNA will recognize the 2 target sites on both sides of the 3PA. It will make 2 cuts and get rid of that 3PA poly A terminator, and then will let the reporter to show M-scarlet color or luciferase activity. So luciferase is an advanced version of bioluminescent imaging system. We have been using this in the group in both cell culture and the mouse models. It's very sensitive and can be better qualified. So we started this project with 3 guide RNAs. One is targeting intron 9, the second one is intron 10. We also have one targeting exon 11. And each of them has in silico or target score as shown here. And I transfected the cells with a bunch of negative controls and our reporter, duplication reporter with each of those guide RNAs. As you can tell from the top, those are the IVIS imaging showing the luciferase activity with the intensity of those colors, and those values were quantified in bar graph as shown below. So our reporter system is really good, and the activity shows, the last one is our positive control. So our reporter shows that the result very well matches the in silico predictor, as shows the intron 10 guide RNA has slightly higher efficiency than the other 2. And those are the microscopy images of co-transfection. Again, if you only put the guide RNA, it will show the EGFP, but if CRISPR-Cas worked in the cells, it will show scarlet color. The top panel I put a duplication reporter plus Cas9 guide RNA, intron 9 guide RNA, and it shows EGFP because the guide RNA is there. It also shows scarlet because the CRISPR-Cas worked. And the second panel is our positive control, which is Cre-LoxP. It's more efficient. You can see more scarlet there, but there's no EGFP because Cre plasmid does not include any EGFP. And the last one is just a negative control. If you only put guide RNA in, there won't be any scarlet showing. So the reporter system worked really well in the KCO2 cells, and that's where I started. We optimized all the conditions in a cell line. Then we applied the similar method in human primary nasal epithelial cells. Those are actually the patient cells we obtained. Everything worked as well as in the cell lines. Our guide RNA can be efficiently delivered into the cells, and cells, they were happy, they can survive the whole process. Now those cells are in culture, and so up to now, we know everything worked, but from the genomic level, so how do we know if those guide RNA can make the deletion as we wanted? So I did, it's a very simple assay. I put 2 different guide RNAs, one targeting intron 9, the other one is targeting exon 11. So the intron 9 to exon 11 is about 14 kilobases. I have primers, one upstream, the intron 9, the other downstream is exon 11. You don't expect to see any PCR product for 40 kbs if there's no deletion made. But if you make a deletion there, you will see a PCR product. And I did a Sanger sequencing. The result shows part of my sequencing came from intron 9, the other part from exon 11. That means all the 40 kilobases are deleted by 2 different guide RNAs. So every pieces work, and this is my little summary that duplication reporter can provide to test our candidates guide RNAs for targeting duplicated regions. And those candidate guide RNA can be efficiently delivered to human primary nasal epithelial cells, and they can make the deletion and restore normal to the normal CFTR gene. So when we obtained the cells from patient and her parents, we performed a short circuit current. Those are raw data. The blue line shows parent, trace parent, and the black one is patient cells. And you can tell that parent cell, they respond to all those manipulations. If you add force clean, it drops and it increases, and if you put the I-172, it drops. But the patient cell, the bottom one, the black line, you don't see any change or very minimum change there. Now the cell, we have the cells, so this will lead our future direction. We have the patient cell, we did gene editing, and now they are ciliated, and right after this conference, we will perform our functional assay on the patient cells. We will see, so we know we can delete the extra copy in that region. If we can, now we want to see if we can restore CFTR function. And if we can, how much we can restore CFTR in the patient cells. And also the second step we want to do, because the region is really big, there are many SNPs in there, you can always find a guide RNA which only target the duplicating allele but try to keep the other allele intact. So we are planning to test more guide RNAs which only target duplication alleles. And lastly, as I said, there are many duplications in CFTR gene. We want to apply this strategy on other CFTR duplication mutations. And lastly, I want to thank my colleagues in Ms. Drum's lab and also Debbie Corey in Tom Kelly's lab, who has been helping me with primary cell culture, and she tried very hard to make those cells happy. And questions? Sorry. I can't hear, I'm sorry. Expansion. When they were in plastic expansion, not in the differentiated cells. Yes. Yeah, we sorted the cells, the positive cells, and after the guide are delivered, then they are differentiated in the filters. Right, yes. Now we just want to see if everything works, then eventually we want to, yeah, like you said, we'll be trying to target in the differentiated cells, yeah.
I have a question. I'm not sure if I missed that. What is the impact of this duplication on the protein? Did you?
Oh, that's a very good question. So this duplication is like you said, 5 exons are involved. Honestly, we don't know how that affects the messenger RNA. If all the exons are included, it will be a frame shift, but you never know if it messed up splicing or other things. So right now, we don't know what protein level. Even messenger RNA level, we are not quite sure, but we just know there's no CFTR function.
So you didn't run the Western for this patient, did you? Run the Western?
We don't have very good luck with Western in human cells. Yeah, we will.
Okay.
Yeah, thanks.
I also have a question. Do you think it will be possible to combine guide RNA for different duplication, or do we need to develop one therapy for each duplication?
I think one of my slides showed that there are a lot of duplication. They have overlapping regions. If one is good for one, should be working for the others. For all those, like I think I showed 10, maybe we only need 4 guide RNA to targeting all of those duplications. We don't have to design for every single duplication. They have overlappings.
Okay, thanks.
So now we'll move to the last seminar of this session. So this presentation is by Catherine Foy, and she's going to present hyperglycemia contributes to oxidative stress and impairs potency of highly effective modulator therapies.
All right. Hello, my name is Catherine Foy, and I'd first like to thank Drs. Aurora and Piedamonte for allowing me the opportunity to give this presentation, and highlights of the findings presented here will also be available at poster number 651. My disclosures for this presentation include funding from the CFF and the NIH. Cystic fibrosis-related diabetes is the most prevalent comorbidity in individuals with CF. And the CFF patient registry from the 2020 annual data report shows that an estimated 30 to 50% of CF adults are diagnosed with CFRD, which develops during adolescence and leads to a progressively worsening prognosis through life. CFRD is characterized as systemic hyperglycemia due to a reduced insulin response or delayed insulin response, as well as insulin resistance. And although CFRD shares some features with both type 1 and type 2 diabetes, its pathophysiology and clinical manifestations are distinct. And one of these distinctions is that mortality is due to progressive lung disease and respiratory failure instead of heart attack or stroke. And significantly, the risk of mortality is increased around 6-fold in CF adults with CFRD. And so improved understanding of the mechanisms by which systemic hyperglycemia contributes to accelerated pulmonary decline represents a significant unmet medical need. And CFRD pathogenesis is complex and multifactorial. Major drivers include CFTR variant severity, inflammation, and oxidative stress. In CF, redox poise is dysregulated from the earliest stages of disease, as is evidenced by the diminished levels of the antioxidant glutathione. Dr. Anthony Kettle's group, in association with Arrest CF, showed this by comparing glutathione levels across 3 oxidative species in pediatric non-CF and CF bronchoalveolar lavage fluid samples. They showed that the reduced form of glutathione, or GSH, as shown here, as well as oxidized forms GSSG and GSSP, were all significantly reduced in the CF cohort as compared to the non-CF cohort. And these results indicate that glutathione is low in individuals with CF from an early age, irrespective of oxidation. And in addition to the oxidative stress already experienced in CF, glucose-induced oxidative stress introduces a double hit of redox imbalance. In this abbreviated schematic of the polyo pathway adapted from Dr. Brownlee, we see that increased glucose triggers aldose reductase to reduce glucose to sorbitol, and in the process that consumes the cofactor NADPH. However, NADPH is also essential for regenerating reduced glutathione. And so by elevated glucose depleting levels of reduced GSH, the polyo pathway increases susceptibility to intracellular oxidative stress. Our objective is to elucidate the impact of hyperglycemia on airway redox poise, as well as modulator efficacy. We hypothesize that chronic hyperglycemia depletes GSH levels by increasing ROS and decreasing NADPH, resulting in CFTR turnover by accumulation of advanced glycation end products. We hypothesize further that our new highly effective modulator therapies, or HEMTs, will stimulate apical secretion of GSH by pancreatic ductal and airway epithelia, thereby improving the CFRD molecular phenotype. Overall, we aim to determine the impact of HEMTs on mutant CFTR biogenesis, glucose metabolism, and redox poise in cells altered by the CFRD sequelae. To study this, polarized cells were grown in normal glycemic conditions, which we determined to be around 100 milligrams per deciliter of glucose, as well as hyperglycemic conditions, which we determined to be around 250 milligrams per deciliter of glucose, and with or without chronic treatment of modulators, which were at a 5 micromolar working condition. And our glucose conditions were based on physiologically relevant levels considered normal or diabetic. CFTR short circuit currents, immunoblots, and GSH redox state were quantified. So So in order to investigate the way in which hyperglycemia impacts lung function, we worked with our collaborator, Dr. Niall McCarty's lab at Emory, to test CF bronchial epithelia, or CFBE, stably transduced with wild type or delta F CFTR. These cells were grown for 5 days under chronic normal or hypoglycemic conditions. And these are the short circuit current measurements from the wild type CFBE. In black is the forskolin response, and in gray is the CFTR specific inhibitor response. And here we see that hyperglycemia alone decreases transepithelial ion transport by around 34%. And a similar trend was seen in the delta F cells, with hyperglycemia conferring around a 33% reduction in CFTR function. We next cultured our delta F CFBE for 5 days with sustained normal or hyperglycemia and 48-hour application of the HEMT, elexacaftor, tezacaftor, ivacaftor, hereby referred to as ETI. And so ETI restored delta F activity to around 34% of wild type, although hyperglycemia did significantly decrease the HEMT response, which was around 31% of the normal glycemic condition. And now immunoblots of these samples show that chronically elevated glucose did not significantly alter delta F biogenesis, either with or without ETI treatment. So while ETI-mediated rescue of delta F is reduced in hyperglycemic conditions, the amount of protein is not significantly altered between normal and elevated glucose levels in the presence or absence of modulators. This indicates that there is a separate pathway being impacted by the hyperglycemia, which could support our hypothesis that CFTR turnover is being accelerated. So in an endeavor to begin to understand the effect of hyperglycemia on glutathione levels, we worked with our collaborator, Dr. Joshua Chandler at Emory, who used LC-MS to determine intracellular GSH levels under normal and hyperglycemic conditions on 16 HBE cells endogenously expressing wild type and delta F CFTR. We found that following 4 days of glucose treatments, wild type intracellular GSH was reduced in the presence of hyperglycemia by around 21%. And the same trend was observed in delta F, although it did not reach significance. And while intracellular glutathione is an indicator of the overall oxidative state of the cell, GSH antioxidant activity is most essential in the epithelial lining fluid of lungs. It has been hypothesized that CFTR may be involved in the transport of GSH across the cell membrane. And because of this, we wanted to see if a modulator treatment would increase apical GSH levels. To do this, we looked at fully differentiated primary human bronchial epithelia, homozygous for wild type or delta F CFTR, treated 8 hours with a control vehicle DMSO, the corrector lumacaftor, or the corrector potentiator combination lumacaftor-ivacaftor. We found that the combined application of LUM-IVA increased apical fluid GSH around 2-fold in delta F homozygous HBE, although there was no significant effect by the corrector alone. This provides evidence that CFTR modulators are effective at improving apical GSH. However, the mechanism of GSH transport is still unknown. And there are important considerations for knowing the effects of chronically elevated hyperglycemia, not just in the airway, but also in other tissues affected by CFRD. For this reason, we tested similar conditions in pancreatic ductal epithelia. In this study, we obtained the PANC1 cell line, which is pancreatic ductal epithelia that endogenously expresses wild-type CFTR, as well as the CFPAC1 cell line, which is similar but endogenously expresses the delta F CFTR. And based on our literature searches and previous airway data, we hypothesized to see similar results in these pancreatic ductal epithelia in regards to hyperglycemia and HEMTs as we saw in our airway cells. We began by doing similar short circuit current investigations on the PANC1 cells as previously done on the CFBE cell line. We cultured the PANC1 epithelia at liquid-liquid interface for 6 days under chronic normal or hyperglycemic conditions with 48-hour vehicle or ETI treatment. And these cells show a similar trend as seen as airway cells for HEMT-mediated current increase in both glycemic conditions. Interestingly, we did not find significant change in transepithelial ion transport between normal or hyperglycemia at baseline. However, the modulator response was attenuated by elevated glucose levels, which is a similar trend to what we observed in airway epithelia. We also recorded an ENaC activity in these cells as evidenced by an amiloride response. We observed that hyperglycemia alone reduces sensitivity to amiloride, which indicates that ENaC is being negatively impacted by the increased glucose, similar to CFTR. This is surprising because in airway epithelia, ENaC and CFTR activity are inversely related. Also, ETI did not have a significant effect on ENaC function in either normal or hyperglycemic conditions. When looking at the HEMT responses of transepithelial ion transport in CFPAC1 cells with the delta F CFTR under strictly normal glycemic conditions, we do see a significant response to ETI. However, it is important to note that the CFTR rescue is around 2 to 3-fold of the vehicle control. In airway cells, we observe around a 20-fold increase in HEMT-mediated CFTR rescue with ETI. So while ETI has been shown to have a significant effect on airway delta F rescue, it remains to be determined if it is similarly effective in other tissues and organ systems. Finally, we evaluated CFTR abundance in CFPAC1 monolayers grown for 6 days in normal glycemic or hyperglycemic conditions with DMSO or ETI treatments. Our densitometric analyses show that ETI application under normal glycemic conditions appeared qualitatively to enhance band B expression, although the quantitative analyses did not indicate statistical significance. And under hyperglycemic conditions, the amount of band B appeared to be reduced. However, again, our quantitative analyses did not prove significance. And interestingly, we found that hyperglycemia alone did not significantly diminish band B expression in delta F, which is in contrast to observations in CFBE cells. And so taken together, our results indicate that in lung epithelia, hyperglycemia impairs CFTR ion transport, HEMT rescue of mutant CFTR activity, and cellular redox state. We were also able to show that modulators do augment CFTR-dependent redox buffering. And we were excited to begin working with pancreatic ductal epithelia to elucidate the impact of hyperglycemia and HEMTs in other tissues, and we're preliminarily able to show that the pancreatic ductal cells are mildly sensitive to HEMT-mediated CFTR rescue and modestly susceptible to hyperglycemia. And further investigations are needed to determine the impact of glucose toxicity and HEMTs on redox state. And as we continue to discover the links between hyperglycemia, CFTR variant severity, oxidative stress, and the interplay of these with highly effective modulator therapies, we will be able to strengthen precision medicine-based approaches to CFRD complications. And we recognize that there are still many pieces left to this puzzle, and we plan to use primary human airway epithelia to evaluate the consequences of hyperglycemia on CFTR functional expression and HEMT response, similar to what was done in airway and pancreatic cells. We are also working towards elucidating the mechanisms by which chronically elevated glucose depletes GSH and NADPH levels in airway and pancreatic ductal cells. And finally, ascertaining the impact of hyperglycemia and HEMTs on glucose metabolism. And I'd like to thank my colleagues in the Catherine Oliver lab at Emory, as well as our collaborators in the McCarty and Chandler labs, as well as our collaborator at Erasmus. And I'd like to take any questions. Right, we, as far as I know, have not checked the cyclic AMP levels yet. but that's something we can certainly do moving forward. I'm sorry. Yes, right. So did we test it in hypoglycemic conditions as well? No, that is another thing that we have not done yet, but that's a really interesting observation and something we should definitely look into moving forward. Thank you.
Any other questions? I have one question. So do you think there is any impact of vascular glucose exposure versus paracrine in the pancreas glucose exposure? Have you tried something with the co-culture system?
Is there a difference in...
Whether that influences the CFTR response?
Oh, interesting. No, we haven't considered that either, and so that would be an interesting thing to look forward to as well. Oh, sorry. We have not looked into the vascular in the pancreatic cells, the vascular CFTR. Or, sorry, can you repeat your question?
Yeah, so the effect of glucose to the pancreas would be paracrine versus the vascular exposure in the airways. So do you think that would explain any differences?
Certainly, I think that could be an explanation for the differences that we're seeing. And our pancreatic ductal epithelial data is very preliminary, and so we're still working to make it a little more robust and really see some of those differences in the airway cells as well.
Thank you very much. So this concludes the workshop, and a round of applause for all the terrific presenters today. And your audience is greatly appreciated, and I hope you enjoy the rest of the conference. Thank you very much for coming today.